time is yours, Dr. Radiana.
Thank you very much, Mr. Chairman, Dr. Ismail. I would like to thank also the committee for the invitation. So I hope that you can learn one or two things about somatic cell nuclear transfer and iPS technology. As an outline, I will briefly mention about the somatic cell nuclear therapy technology then and now. I will speak about iPS technology, how it begins and how it develops until now, and whether it's applicable, iPS technology, for Indonesia's current stem cell research. And I would also briefly mention about how to go forward with the stem cell research. If you see this cartoon on the right side, it reminds us all on our beginning. So we start from a single cell, which is a zygote, which proliferates into morula, blastula, and in the inner cell mass of a blastula is where you can find embryonic stem cells. These cells are pluripotent, means it can be differentiated into over 200 types of tissue in our body. And then we continue our development into postnatal as a baby and to adult and to aging or elderly person. Actually, we still have stem cells in our body. They are residing in each organ, but the number keeps dropping with the advancing of age. With the somatic cell nuclear transfer, this paradigm is shifted. People now can reprogram their cells back into younghood, so they can be totipotent cells or pluripotent cells. Recently, Yamanaka introduced a technique called induced pluripotent stem cells, which is an iPS cells, and I will talk about Yamanaka's finding later on. So we start with the somatic cell nuclear transfer technology. So if you still remember Dolly as a sheep clone in 1997, it makes a highlight in Science. So it's a method to reproduce, you can say, organism, a whole organism from a single cell. So usually it is used in a cattle to improve the breeding or the quality of the sheep or other cattle. It is once thought that by this technique, by somatic cell nuclear transfer, you can use the cell for therapeutic cloning. What does it mean? It means that once we have an inner cell mass of a blastocyst from the reprogrammed cell, so it started from an ovum gets its nucleus removed. It can be by electroporation technique, or it can be from other technique to dissipate the membrane of the cells, and then you introduce the nucleus from other adult terminally differentiated cell. And then the cell will obtain the same genomic composition as its parent or as its donor nucleus. And then it will be proliferate further to morula, blastocyst, and then where we can obtain the inner cell mass. It is thought that from this pluripotent cell, we can differentiate many tissues and we can give it back to the patient. However, this technique is very much less success. There has been a report on making pluripotent cells with the somatic cell nuclear transfer, but none of them ever reaches differentiated cell therapy for patient. Yamanaka, together with Sir John B. Gordon in 2012, actually give an idea or illustrate the milestone until his finding of induced pluripotent stem cell techniques. And as you can see, it started with the John B. Gordon experiment on how he removed from the ovum with adult intestinal cells from a frog, and he can clone a frog. So he gave an idea that an adult terminally differentiated cell can be rejuvenated back and clone a whole organism. Ian Wilmut picked up the idea in 1997, and Weintraub in 1987 said using a single transcription factor, which is a MyoD, a person can differentiate skeletal muscle tissue. Tada, in 2001, picked up the SCNT methods by fusing an ovum with embryonic stem cells, and he can achieve pluripotent cell. And also, if you want to look at other milestone regarding the embryonic stem cell isolation and culture, it ends up to the finding of the technique. So how does it done? How is the induced pluripotent stem cell being produced? This is a schematic showing you a very simple linear approach. So first of all, we need to take out differentiated cells from the patient or from an adult. For instance, this is the fibroblast from the patient. And then we transduce with a virus containing 4 transcription factors. As you can see, OC4, SOX2, NANOC, and the other one is KLF. With these 4 transcription factors, the cells can then be selected. The clones, which is previously sensitive to neomycin, can be selected based on neomycin resistance, and these cells are partially reprogrammed. With further culture, these cells can induce its own endogenous expression of OC4, SOX2, and NANOC. And when that happens, it means that the cells are completely or fully reprogrammed into its iPS state or pluripotent state. And the virus expression is already silenced. So actually, this is thought to be a quite simple and straightforward methods. So this is Yamanaka's first paper in 2006 in Cell. So Yamanaka uses this vector construct, and he uses with beta-galactosidase. The promoter that he targeted is the FBX15 promoter, and there are initially 24 genes that is being screened. Afterwards, only 4 transcription factors remain, which is OC3-4, KLF4, SOX2, and CMYC. And as you can see, this is the type where they selected the clones that is resistant to antibiotics, and these clones can be propagated further, and the number of the yield, the production is quite robust at that time. 5 out of 12 clones survive. This is the morphology of the colony of an iPS cell. It is much similar to the colony of embryonic stem cell in comparison to the natural mouse embryonic fibroblast. So this is the initial cell that is being reprogrammed. And then Yamanaka tried to compare between its iPS clones, depicted here in this white circle, with the ES cell depicted in a black square. And in comparison with the initial or natural cell, the mouse embryonic fibroblast. As you can see, the population doubling time is similar with the ES cell, which means this iPS cell has really gained phenotyping or traits of embryonic stem cells. And also shown in this reverse transcribed PCR, where the investigator of Yamanaka tested several markers of pluripotency such as OCT3/4, SOX2, FGF4, and he found iPS clones to possess some factors of pluripotency as embryonic stem cells in comparison with the native cells. This is actually a DNA methylation result to show the epigenetic condition of the iPS that is produced. As you see, between these two clones of iPS that Yamanaka produced, the black circle denotes methylated CpG island, while the white circle represents unmethylated CpG island. But what you need to see in here is this is the promoter area of the OCT3/4, NANOC, and FBX15, and you can see the pattern are much similar to the native cell in comparison with the embryonic stem cell. What does it mean? It means that the epigenetic memory remains. The cell still have a memory as fibroblast instead of embryonic stem cell. Epigenetic means it's outside the genome, outside the DNA, in the chromatin. If you remember, the genes are compact in the chromatin. And Yamanaka also tested other genes with reverse transcribed PCR. As you can see, also not having a very much success with the pluripotency genes. But this is also can show you that the exogenous gene has been silenced. And they're waiting for the endogenous genes as early as day 10 or passage 10. This is a heat map from a microarray result. And what you can see from this is each line represents the sample. The green denotes down-regulated expression of genes in comparison to control, which means the fibroblasts. And if you look at the heat map pattern between the iPS, they are much more similar to the ES cell in comparison to their native cell. Yamanaka tested the capacity, the differentiation capacity of this iPS clone by injecting into immunodeficient mice, and nicely he can show that there is a tumor formation, and within that tumor, there's a derivative of 3 lineage from the mesodermal lineage, which is the muscle, the cartilage, from the ectodermal lineage, which is the central nervous system, so this is like a neuron, and the epithelium, and also from the endodermal lineage. He also tried to differentiate into other, such as liver, but or liver cells, so this is an early marker of liver cells, which is the alpha-fetoprotein, and he showed nicely that it can be differentiated into other types of cell. But if you want to know how it's done, actually, you need to master these components to reprogram a cell from terminally differentiated back to its pluripotent state. Not only you need to modify the transcription factors, but you need to also regulate the chromatin remodeling. And also, there are target genes that can be silenced using miRNA. So these are really molecular circuitry that need to be mastered to obtain an iPS cells. And I like to show you how the success rate of iPS technology. So if you look at several methods to reprogram adult cell into iPS cell, and most of this data are coming from human cells, the efficiency rate is still very, very low, ranging from 0.01% to 4.4%. So it's a lot of work, low efficiency. But what can we do with them? Can we make it? Can we make this iPS cell and translate it into the clinical area? Yes, we can. We can make a good manufacturing, so clinical good manufacturing product grade iPS cell. How do we do that? It's by performing a culture that is free from all animal proteins. So for instance, if we need a feeder layer, the pluripotent cells cannot be cultured on top of a mere flask. It needs to be coated with something, or it needs to be supported by a type of cell or matrix, and that's why we use they use synthetic matrigel. Other things that they improve is the use of medium, xeno-free medium. So for instance, this is using Syntamax, and they can nicely show that this is comparable, that the iPS cell that they produce is showing the pluripotency factor expression even higher than the embryonic stem cell line. And this is to show, so they transferred this cell from a differentiated cell, and they wanted to get rid of the lineage-specific marker, which is the N-glycosyl neuraminic acid. And they successfully showed that with xeno-free condition, they can achieve an iPS clone without the expression of lineage-specific cells or tissue. So the next question will be, is it applicable for Indonesia? Even Yamanaka stated in his recent review that iPS technology application needs to stay outside of the patient body. It means that it can be used for embryogenesis study. It can be used to study mechanism of disease, in vitro drug testing, or teratology of a drug. So these are the recommended application of iPS. However, in the field of cell therapy, I can show you later on, there are still many challenges. So what I think we could use this iPS technology is to produce. It really depends on where you belong. If you belong in an academic health center, for instance, in Universitas Indonesia, or in other stem cell research center that is based in academic institution, you can generate patient-specific iPS cell. And this has been done. There are over 20 publications already, ranging from ALS to Parkinson to other chronic diseases, and they yield patient-specific stem cells. What do they use this iPS for? It's for in vitro disease modeling and for drug testing. So this is a very good system to test new drugs or to understand the pathomechanism of a disease. And you can use iPS for bioartificial tissue or organ. And you can use iPS for embryology or developmental study. Or if you're in a tropical infectious disease research center, you can use iPS for in vitro testing. to see the mechanism of parasites entering the cells. So it's, you can use that iPS technology. In the research institute as well for in vitro drug testing and for bioartificial tissue or organ. For pharmaceutical company, the iPS can be used for new drug development. So if you have a lead compound and you need a system, a robust system, where the cells can be differentiated into any types of tissue, then you need to go with the iPS. Okay, my time is up. 2 more slides. Okay. So the benefit and challenge is that iPS can be used for bioartificial tissue and organ. And if by any chance you need to transplant it into this immune privileged site, because these sites are avascular. There are 2 sites in our body that is known to be immune privileged site, which is the cornea and the cartilage. So you can transplant iPS in this area. However, the challenges are still low reprogramming efficiency and tumorigenicity. This is one slide that I used to give to my students. Only with 2 cells, 2 pluripotent cells. So if you have stem cell and you differentiate it into type of tissue, you still have a mixture of cell. You can still have pluripotent cell in those mixture of cells. And if you transplant it to the patient with only 2 cells, the patient might form tumor or can have tumor. 40% of the, this is in animal model, 40% of the animal have tumor within 15 weeks or so. So this is a very high concern safety issue. This is my thank you note. So actually, we have performed several work for our roadmap in bioartificial liver. So the first 2 results have been presented in China and in Singapore. It's for the 3D co-culture methods. And below one is other 2 works from peripecial skin. Where do we go next? Where do we go next is we are making this bioartificial liver as a bridge therapy for liver transplant candidate. But from the peripecial skin cells, we won't go into iPS due to the safety reasons, but we will make a direct conversion. So it's like direct programming from peripecial skin cells into hepatocyte. It has been reported as early as 2011. Thank you very much.